Aloha, everybody. The time has come for our last lecture in this course. And this particular topic is perhaps one of my favorite topics because as a young graduate student, I began using RNA interference. At that time, it was this very hot topic and research technique that only few had used. As a matter of fact, it was so um, unpopular. Well, it was popular but unused, and not many people knew how to do it. That I was able to follow and read every single paper that came out, and that in the realms of about a year or two. And I remember that by the time I graduated, which was about 4.5 years later, um, I couldn't follow the research anymore. It was so the journals were so overflowing with publications that I just couldn't follow. I couldn't read all of them, and that's how fast things developed when RNAi was discovered. And it's still widely used, and it has great potential. And as you all know, CRISPR-Cas9 was this recent discovery just a few years back, which holds this magnificent potential for great benefits to all of us, and of course the potential to do a lot of damage and a lot of hurt. Um, Now, our understanding, or let me step back. How do we ascertain the value of these studies? Has to start with understanding of what they are, what these technologies are, what these mechanisms are, what is their potential, what is their power, what can you do with them. But we cannot really know what their potential is without understanding what they are. So in this presentation, I'm going to try to give you just a framework and a foundational explanation of what RNA interference and CRISPR-Cas9 systems are. Now, as a phenomenon, gene silencing driven by microRNA or small interfering RNAs was discovered by Craig Mello and Andrew Fire, for which they did receive the Nobel Prize. MicroRNAs are naturally existing RNAs in the genome. They have the ability to silence or inhibit gene expression in plants and animals, in bacteria as well, in everything, and in a living organism. MicroRNAs do their function by interacting with mRNA. By interacting with the mRNA, they either lead to inhibiting the translation that is based on that mRNA or the complete degradation of the mRNA. Either way, the translation or the final gene expression product will be decreased. It will not be completely removed. It will just be decreased to a certain level. Might be just 50%, might be 80%, might be 90 or 95%. Either way, we're talking about knocking down expression or inhibiting expression and not completely deleting the expression. A natural question would be, what could be the physiologic effect of this process? Why do our cells have these mechanisms? Well, one of the beautiful things of this mechanism is, like I mentioned, is that this is a temporary regulatory mechanism. In other words, we're decreasing the expression for a certain amount of time. It's not permanent. after we control the expression of the microRNAs, we can get that particular gene that matches the mRNA to express again. So that's why this is so beautiful, because you have a certain, well, the mechanism is transient. You have malleability. You can have in one cell have higher expression, in some of the cell lower expression, in some of the cells lower to a minimum expression. And that matters for what that cell will become or what that cell will do. mRNAs usually interact around the 3' end of the microRNAs usually interact with areas in mRNA that are close to their 3' end. It's not true for all of them, but it is the most common location of the interaction sites are close to the 3' end. The additional roles for the microRNAs is that, well, I shouldn't say additional, but basically to generalize what I said is their role is in development, in tissue profiling, in defending against invading RNA, viruses especially, and it's noteworthy to say that there are hundreds, perhaps thousands of microRNAs in the cell. This form of regulation is of incredible importance for development and tissue profiling. And now when we talk about invading viruses, this is a defense mechanism against viral infections as well. Now, in some cases, it's not very successful, but in some cases, it is. All right, now, microRNAs as entities are not just simply synthesized as microRNAs. The mature or functional microRNAs are about 20 to 25 nucleotide long hairpins. They're just small hairpins. And it's been estimated that approximately 1,500 genes express microRNAs. And again, these microRNAs can regulate various genes. They don't have to regulate the gene that expresses them. They can, but they don't have to. But they can regulate genes that are completely unrelated to the gene that expresses them. They're initially synthesized as a much larger precursor that is known as the primary transcript for microRNAs, or shortly put, mRNAs. It is, well, the locations for expression of these microRNAs are found in various parts of the genome, but it is not uncommon for them to be located within introns of genes that either they regulate or genes that they don't regulate. Regardless, I've made this point a little bit, well, in one of my previous presentations, is even though the intron will be excised and it's considered unimportant for the protein expression, these introns are not always unimportant for the cell, because they contain expression sequences that code for regulatory RNAs such as microRNAs. Now let's go over the mechanism of action of microRNAs, and particularly the synthesis. All right, you can see on this image, everything is described in this image, that in the nucleus, the mRNAs are synthesized, and they can have various lengths. So you can see them how they are synthesized by RNA polymerase II. The mRNAs are digested by drosha and DGCR8 to pre-microRNAs, which are about 70 to 80 nucleotide long. Then the pre-mRNAs are transported to the cytosol in a complex with exportin 5 and RAN, and with the help of GTP. With the help of dicer and helicases, the pre-mRNA is converted to a mature RNA. The mature RNA is basically, as you can see, has a single-stranded element to it, which is obviously going to be important in recognition. Now, the mature microRNA is incorporated into the RISC complex. RISC stands for RNA-induced silencing complex. Now, the RISC complex, with the help of the microRNA, binds to target mRNAs that have complementary sequences. These sequences can be, can be near perfect. The complementarity could be near perfect, or they can have one, two, or three nucleotides that are not complementary. Based on the level of complementarity, if we have partial complementarity, we have transcriptional repression. In other words, the mRNA is not destroyed, we're just suppressing the translation driven by or based on this mRNA. If the match of the mRNA with the RISC complex and the microRNA is near perfect, that mRNA oftentimes is being degraded completely, and as a result, we have a decreased expression that is based on this mRNA. Now, there is, this process that is utilized by the mRNA is referred to as RNA interference, or RNAi. Some microRNAs work only on one gene. Some microRNAs can work on multiple genes based on their sequence similarities. In plants, this was particularly discovered in plants, but it also works in other cells. When a double-stranded RNA, which normally comes from viruses, enters a plant sequence or another mammalian sequence, that long double-stranded RNA is being digested into small double-stranded RNA segments that are about 21 to 23 nucleotide long. These small double-stranded RNA segments are known as small interfering RNAs, or siRNAs. The siRNAs are then incorporated into the RISC complex that leads to the silencing of the complementary RNA or gene. And again, this process is known as RNA interference. On this side of the figure, we see this example where we have imperfect matching of the double-stranded RNAs, and these are called small temporal RNA, and they're involved in silencing as well, similarly to the siRNAs. This process is used successfully in virtually any organism: plants, invertebrates, mammalian cells as well. Now, the mRNAs, together with these other mRNAs, are called non-coding RNAs. And it's been found that mammalian genomes have more non-coding RNAs than coding RNAs. Just for your information, SN stands for small nuclear, and they work with nucleosomes. The SNO means small nucleolar, that are involved in the processing of ribosomal RNA and other non-coding RNAs. L and C are long nuclear RNAs. rRNAs, obviously you know what it is, tRNAs know what it is, and NC stands for non-coding RNAs. All right, so this is RNA interference. I will come back with the CRISPR-Cas9 in a few minutes. Welcome back, everybody. We will finish our lecture today with discussing the CRISPR-Cas system. CRISPR stands for Clustered, Regularly Interspaced, Short Palindromic Repeats. Cas means CRISPR-associated nuclease. So basically, from now on, when you hear CRISPR, you should think about clusters of palindromic sequences that are separated from each other by spacer DNA. That's what basically CRISPR means. Clusters of palindromic sequence repeats that are separated from each other by spacer DNA. We'll talk about it a little bit more later. Cas is basically a nuclease that is associated with these CRISPR sequences. The system as a whole, acting together, they form a defense mechanism that was discovered first in bacteria, and it's designed to help them protect themselves against bacteriophage infections. So what is the mechanism of that defense? It is a natural mechanism. It's a naturally occurring mechanism that we now, after we understood it, we now are trying to use it to see if we can do gene editing for higher organisms. So the bacterial genome normally has these palindromic repeats. They're clusters of palindromic repeats. However, when bacteria gets infected by bacteriophages that have their own viral or bacteriophage DNA, when that DNA enters the bacterium, it also has the ability to incorporate itself into the bacterial genome. And that incorporation often happens within these palindromic repeats, these clusters of palindromic repeats. And what basically happens is you have a cluster of palindromic repeats spaced by these viral insertions, and then followed by another cluster of these palindromic repeats. In essence, you get these clusters, like I said, of palindromic repeats that are spaced with inserted viral DNA. These complexes, these sequences, are known as CRISPR sequences. Now, when a phage attacks for a second time, the existent CRISPR sequence that already has the previously incorporated viral or phage DNA, that existing CRISPR sequence will be transcribed to RNA. When that RNA is generated, the single-stranded RNA will serve as a guide to base pair with either DNA or RNA of the bacteriophage that attacks the bacteria for a second time. That RNA serves as a guide, and it's known as the guide RNA. The guide RNA, together with another RNA called transactivating CRISPR RNA or tracker RNA, helps to recruit the Cas nuclease and other nucleases that then will destroy the phage DNA. So that's how it works. The first insertion of the viral or the bacteriophage genome within the bacterial genome will serve as a template to express RNAs. These RNAs will serve now as a guide to recognize second or third invasion from a bacteriophage to that bacteria, and with the help of a tracker RNA that recruits the nucleases, the guide DNA and the tracker RNA and the nucleases together will recognize the foreign phage DNA and will destroy it. So that's, in a nutshell, how it works. Now, the CRISPR-Cas9 system is more specific. It was first observed, and it exists in Streptococcus pyogenes. It is simpler in the fact that it only has one nuclease, Cas9. This phenomenon was discovered and developed by Jennifer Doudna and Emmanuel Charpentier, and they use this technology and utilize it for correcting or deleting genetic information. The best word is for editing genetic information. So this technology that was observed in bacteria, now we're trying to use it to edit the genomes of higher organisms. What is the mechanism? How does it work? How can we utilize that technology? And also a good question would be, what are the implications? There are a lot of discussions going on even now of the ethics of this discovery and the potential of this discovery to open doors for many improper reasons for using this technology for cloning or for editing of human genomes and for generating traits that are perhaps desired by some, but maybe not ethically desirable. Okay, so let's focus now on the science of things and leave the politics aside. Okay, so like I said, this is the technology. This is something that normally does not happen in a human or mammalian cells or genomes. We generate a plasmid, that is CRISPR-Cas9 plasmid, and we also have a recombination fragment. That recombination fragment will be used only if we want to do our recombination editing. If we don't care of what exactly is going to happen with the gene, then we don't have to use it. Either way, we prepare CRISPR-Cas9 plasmid. That plasmid will be able to deliver the guide RNA, the tracker RNA, and the Cas9. They are now put into cells and they express. When they express, the guide RNA, which is shown right here, that will do the recognition, is fused with a tracker RNA that forms a single guided RNA. So the sgRNA, which is single guided RNA, basically is a fused product between the guide DNA, which is about 20 nucleotides, and the tracker DNA that will be recruiting the Cas9 nuclease. Okay, so the single guided RNA together with Cas9 and other nucleases that are part of the cell will form a complex. Then Cas9 will have the ability either to cut one strand or two strands with its domains. Then the complex will start scanning the DNA until it recognizes the target sequence. If both strands of DNA are cut, the repair now of that DNA will be done by non-homologous end joining, which usually adds or deletes nucleotides, which will change the frame and essentially deactivates the gene. It neutralizes that gene. Even if you add 1, 2, or more bases, it will neutralize the gene because of the deletion or the addition of nucleotides. Unless you add only 3, which is basically going to change, it won't change the frame, it's just going to incorporate an amino acids. But either way, most of the time what happens is that the two strands are cut, and then we have a non-homologous joining, which usually deletes or deactivates the gene. If one strand is cut, shown right here, Now, the recombination fragment that we started with can be utilized, and via recombination can exchange and repair the defect. At this point, this is very inefficient, but it does happen. It has these recombination sites right here that allow for the exchange of what is between them. Basically, this part of the DNA will come out, and this red portion of this on this schematic right here will replace the blue part. You can use this technology to add a base, controllably, of course. You can add a whole protein domain. You can add a whole sequence of a whole protein. I mean, the smaller the better. But the fact that we can exchange DNA and repair DNA, the power of this is incredible. Deleting a DNA, it's very incredible and amazing of itself as well, but we have other ways of knocking out a gene. But this portion, this part right here, where we can repair certain areas of a gene or an entire gene, hold an incredible potential for this technology. And that's the part that is most exciting and at the same time the scariest. So what are the implications? Well, this technology, like I said, can be used for many things. Of course, we as scientists and as normal human beings think about gene therapy. There's so many diseases that are based on faulty genes. If we can use this technology for controllable excision of faulty genes, even at the smallest stage of human development, we will be able to have healthy babies being born with repaired genes without cystic fibrosis or without diabetes or any other mutation diseases that are based on mutations. However, more ambitious people think about adding traits to the human body or just simply making somebody, like some people say, superhumans. I'm going to leave that to you guys to think about and to politicians to talk about. I wanted to point out your attention to these videos that I've included, these links. The first link, it would let you see, well, will guide you to see basically what I just said, but taught by Jennifer Doudna in iBiology. She basically explains the CRISPR-Cas9 system probably, well, not probably, much better than I do. You can also watch her TED Talk in London, where she talks about this technology and the potential use of it. There's also another video from Nature Methods that focuses more on the methodology. And the use of it and the implications is also discussed well in this video prepared by the MIT Technology Review. So if you want to learn more about the CRISPR-Cas9 system and its potential use, you should see these videos.